So let us start the drug resistance, or particularly the antimicrobial resistance. And this part is particularly concerned to the chemotherapy. So this is very interesting that if you are going to treat a patient which are having the infection, and the infection is from the resistant variety of microorganism, then this become very tough to treat the patient, because the bacteria is resistant or microorganism is resistant. For that, you have to select a particular antimicrobial, and that will enhance the price of the antibiotic. Additionally, for that, you have to go through the test like culture and sensitivity test, as well as you have to go through the longer time of treatment. And the best example suited in this is tuberculosis. So if tuberculosis bacteria become resistant to antimicrobial agent, then this become very tough to cure the tuberculosis, because it required drug, you have to pick the drug, as well as you have to go through longer time of treatment. And of course, the finally it will leads to the most costlier process. Likewise, the other complication because of the resistance problem is that in 2016 a report came that about 4,90,000 people develop multiple drug resistance from tuberculosis, and because of this resistance problem, the cost for treatment is not only costlier, as well as the patient become quite susceptible to HIV and malaria, as well as its treatment simultaneously become somewhat tough. So this is very important to know the drug resistance. To check the occurrence of drug resistance, we have to go through the judicious use of antimicrobial. So let us start with the what is the drug resistant or bacterial drug resistance. So when you are giving any antibiotic to kill or halt the growth of the bacteria, the maximal level of the antibiotics that is tolerated by the patient, and even though bacteria are not going to killed, or growth can be halted or checked, then we can call the bacteria become resistant. Again, I am going to repeat the same theme. If you are using maximal tolerated dose of any antimicrobial agent in a patient, at that dose the antimicrobial agent are not able to kill the bacteria or halt the bacteria or check the growth of bacteria, is called bacteria become resistant. So this is very important. Some of the bacteria are inherent or genetic, or you can say this inherent resistant. What is inherent resistant? That is, the bacteria is not resistant by other means, but because of some problem with the antimicrobial agent to enter or to reach up to the target, the bacteria are not going to be killed, and therefore we call that the bacteria is inherent resistant to the antimicrobial agent. Very good example of this one is penicillin G, or you can say the penicillins. Penicillins are usually, in majority of case, particularly the penicillin is active against the gram positive bacteria. However, the gram-negative bacteria are inherent resistant to the penicillin G because the penicillin G is going to kill the bacteria by inhibiting the synthesis of phospholipid. Uh, that is called penicillin binding protein. So this is going to inhibit the cell wall synthesis. Sorry, cell wall synthesis. It check the cell wall synthesis, and major ingredient of this one is the phospholipid. So the content of phospholipid in gram-negative bacteria is meager, is less in amount as compared to gram-positive bacteria. So the target amount where the antibiotic is going to act is very meager in amount. Therefore, the gram-negative bacteria is inherent resistant to the penicillin G. So this is important, as well as gram-negative organism, the penetration of drug or antimicrobial agent are somewhat different from the gram-positive microorganism. So how this resistance will produce in the bacteria, that we are going to discuss. So the bacteria become resistant, I mean a susceptible microorganism, or you can say sensitive microorganism to an antimicrobial agent become resistant strain. How a susceptible microorganism become resistant one? After mutation in genes, or this may be because of acquired resistance, or this may be through selection. So these are the protocol through which a susceptible bacteria, a sensitive microorganism or bacteria become resistant after the mutation or after acquiring the resistance property from the other resistant bacteria or through selection. So now we are going to discuss what are the mechanisms through which a bacteria become antibiotic resistance. So we discussed that. So you can say this is usually by two ways. A susceptible bacteria, a sensitive bacteria become resistant to the antimicrobial agent or antibiotics. How? That we already discussed, because of the mutation, and that we are going to discuss the genetic alteration. So if genetic alteration, genetic sequence changes occurring in susceptible microorganism, the susceptible microorganism may become resistant to the antimicrobial agent or drug resistance, you have to call it. And this mutation may be through a spontaneous mutation, where the mutation occur in the sequence of the gene, in the sequence of the DNA, the sequence get changed. So therefore, because of the changes in the sequence of the DNA, through the mutation, through a spontaneous mutation, a susceptible microorganism become converted into the resistant, or it may be because through the DNA transfer. What it means? Transfer of genetic material like DNA from a resistant bacteria to a susceptible bacteria or to a sensitive bacteria. These all are coming under the genetic alteration in the DNA through a spontaneous mutation or by acquiring it from the other bacteria from the environment. Second one is the alteration of proteins, means ki altered expression of protein in drug resistant organism. Means ki resistant organism get changed, particularly the proteins. So this mechanism is divided into two parts to express the altered expression of protein in drug resistant organism or resistant bacteria through modification in target site. So what is here the target site? The every antimicrobial or different type of antimicrobial agent have different targets, like sulfonamide having the target of the enzymes that participated in folic acid synthesis. Take the target for fluoroquinolones also having the target like enzyme DNA gyrase. Take the example of the penicillin. Penicillin is having the target of the penicillin binding protein like transpeptidase. This is another enzyme that is the target for penicillin. So every different type of antimicrobial are having different target in the bacteria. It may be present on the cell wall, it may be present in the nucleic acid. So the target get changed. So if the target of the antimicrobial get changed, the inhibition of growth of the bacteria, the killing effect of the antimicrobial over the bacteria is not furnished, not completed. So therefore the drug is not going to work to kill the bacteria. Another mechanism regarding the altered expression of protein in the resistant bacteria is decreased accumulation. What it means, decreased accumulation means कि if a target of antimicrobial agent is inside the nucleus, so the antimicrobial has to reach within the cytoplasm by penetrating the cell wall. So if a resistant bacteria by any means checking the penetration of antimicrobial agent to reach to the target site in the cytoplasm, it will check the activity of antimicrobial agent. How? See the fluoroquinolones. Fluoroquinolones target is DNA gyrase, and this DNA gyrase is present or participating in the replication of DNA. So this DNA gyrase is present in the nucleus while the doubling the number of DNA. So the drug has to reach in the cytoplasm. So when the bacteria has developed any such type of property by which bacteria is either inhibiting the entry of antimicrobial agent or either efflux it out from the cytoplasm to outside of the bacteria, and therefore it develops the resistance. Take the other example, ribosomes. The main ribosomes is protein synthesis. So many antimicrobial agents are inhibiting the protein synthesis. So where this protein synthesis occur? That is within the cytoplasm through the ribosome. So bacteria, there are two type of ribosome, 50 and 30S ribosome. So some type of the antimicrobial agent are 50S ribosome inhibitor and some type are 30S ribosome inhibitor. So the drug has to reach in the cytoplasm. So by any means, either through inhibition of penetration of antimicrobial agent up to the cytoplasm is inhibited, or either the drug is efflux out from the cytoplasm to outside of the cell, the drug is not present in sufficient concentration in the cytoplasm, and therefore the bacteria become resistant. Third is altered protein under the altered expression of protein of the resistant organism. What these resistant organism is going to do? These resistant organisms are going to synthesize some enzyme, and these enzymes are going to change or either produce some changes in the antimicrobial. So once there is some changes occur in the antimicrobial agent, therefore antimicrobial agent lost its integrity as antimicrobial agent, and therefore that is not going to produce its mechanism of action. Take the example of the penicillinase or cephalosporinase or beta-lactamase. So these enzymes are synthesized by the resistant bacteria, and that is the protein. And what these enzymes are going to do, that are going to change the structure of the antimicrobial agent. Penicillinase or beta-lactamase is going to destroy the beta-lactam ring of the beta-lactam antibiotics. Therefore, beta-lactam antibiotic lost its characteristic property or identity of antimicrobial agent. Therefore, it lost its activity. So, by these mechanisms, drugs are ineffective to kill, to check the growth of resistant bacteria. Now we have to go through one by one how the genetic alteration occur in the resistant bacteria to produce the drug resistance. I mean, the antimicrobial agents are not going to be effective or showing efficacy against the bacteria. So first is the actually what will happen. Resistant bacteria is going to go through the spontaneous mutation, and because of the spontaneous mutation the sequence of the DNA get changed, and therefore the antimicrobial agent are not having agent are not going to kill or check the growth of the bacteria because of the change in the target, means ki change in the bacteria. So because of the spontaneous mutation there is some changes occur in the bacteria, and therefore the resistant bacteria are going to inactivate the antimicrobial agent. Now we have to discuss this in more in detail. This resistant may be acquired. How? If a bacteria is resistant, I mean their DNA sequence is changed because of the mutation, and actually this genetic changes may be the extra chromosomal or chromosomal. Extra chromosomal changes means ki changes in the sequence in the plasmid, and chromosomal means ki this is the nuclear chromosome DNA sequence get changed. So this resistant bacteria can transfer the genetic material of resistant to another bacteria which is susceptible, and after acquiring or gaining of resistant gene the susceptible bacteria get converted into the resistant one. So that is called the acquired antibiotic resistant. This may be temporary, this may be the permanent. Now we have to go through the spontaneous the mutation of the DNA. This, as we already discussed, this spontaneous mutation of in the DNA may go through or occurring on the chromosome, and this chromosome may be the extra chromosome, that is the plasmid, and mostly the resistant bacteria the mutation occur in the plasmid or extra chromosomal, and somewhat also the chromosomal or nuclear one. So extra chromosomal or plasmid mediated resistant is more happen in the resistant bacteria as compared to the nuclear chromosome. So what will happen? This change the sequence of DNA. How? Either through insertion, either through deletion, either through substitution of one or more nucleotide with the genome. As a resultant, mutation will occur. This mutation may lead to resistant bacteria or may not lead to the resistant bacteria. So once a bacteria become resistant, this become that is not going to be killed by antimicrobial agent against which the resistant bacteria has developed. Take the example the rifampicin or rifampin. Rifampin is a anti-tuberculosis agent, and when we are using rifampin or rifampicin alone or a single antibiotic for the therapy of tuberculosis, then the chances of bacteria to tuberculosis bacteria, mycobacterium tuberculosis, to develop the resistance. So once the bacteria develop resistant against the rifampicin or rifampin, then the bacteria is called rifampicin resistant mycobacterium tuberculosis. And when you are going to administer the rifampin in that resistant against that resistant mycobacterium tuberculosis, rifampin is not going to kill that mycobacterium tuberculosis, and that is become resistant, and that is multiplying there. So this is very very important. Now we have to discuss how these resistant genetic material are going to be transferred to the other microorganism that is susceptible one, that is the sensitive one, or sensitive bacteria or susceptible bacteria. Sensitive bacteria or susceptible bacteria are those bacteria that are killed or inhibit growth inhibition occur by that particular microorganism. So once that susceptible or sensitive microorganism get converted into resistant one, this may be acquiring the resistant genetic material from the resistant bacteria either through horizontal transfer, or you can say the mainly through the horizontal gene transfer. So what is this gene transfer? A resistant bacteria gene is going to be transferred to a susceptible or sensitive microorganism by three processes: transduction, transformation, and conjugation. And these three are called horizontal gene transfer. And as we earlier mentioned, usually bacteria will develop the resistance through the mutation, and that is extra chromosomally, and we are calling it R factor, and that is related to the plasmids, that is extra chromosomally. So that resistant gene should be transferred to the susceptible or sensitive microorganism either by conjugation, transformation, or transduction. This is called horizontal gene transfer. So how this transfer will occur? We have to discuss one by one. So what is transduction? We all are knowing that the transduction is a process in which a phage, a virus, a bacteriophage, a virus is enter into a resistant bacteria, and this bacteriophage acquire the genetic material of that resistant bacteria, and this acquiring of genetic material or DNA by the bacteriophage get transferred to the another bacteria which is susceptible. So what will happen? The genetic material, if transferred by a virus or bacteriophage from a resistant bacteria to another susceptible bacteria, and later on the susceptible bacteria get converted into the resistant bacteria by acquiring the resistant genetic material, is called transduction. And this has been seen in the Staph aureus. Staph aureus acquire the genetic material of resistant Staph aureus by the process transduction. And if you are going to give the antibiotic, earlier earlier that is susceptible one, that antibiotic is not going to halt the growth of the bacteria or kill the bacteria. So now this become resistant one. Another process is transformation. So what is this transformation process? If a bacteria is resistant and acquiring the DNA material and possessing the sequence of resistant property, so what will happen if a resistant bacteria possessing the resistant DNA material died, and after the death the bacteria will remove the genetic material in the environment. Again I am going to repeat the same thing. If a resistant bacteria after its death or by any means DNA material, that is the resistant one, should be transferred to the environment, and that genetic material, the resistant one, is taken by the another susceptible bacteria or sensitive bacteria, then that susceptible bacteria or sensitive bacteria, by acquiring the resistant gene material from the environment, get converted into resistant bacteria, and the process is called transformation. The example is the penicillin resistant pneumococci. So earlier pneumococci was susceptible to the penicillin, but when the pneumococci acquired the DNA material from the environment and get converted into the resistant one, then after giving the penicillin drug to that converted bacteria, resistant bacteria, pneumococci, the bacteria will not be killed by the penicillin. Why? Because in this the penicillin binding protein, that is the transpeptidase or other different enzyme on which the penicillin is acting, is get altered. So when the structural changes occur in the target, then how the antimicrobial is going to kill the bacteria? So this is the another process. Now we had already discussed the transduction, transformation. Now we have to go through the bacterial conjugation. So what is this bacterial conjugation? The bacteria is going to form the conjugate between a susceptible and a resistant bacteria. What will happen if a resistant bacteria get form the conjugation with susceptible bacteria, and the gene material that possessing the resistant property in the resistant bacteria get transferred to the susceptible bacteria by forming a bridge, by forming, that is also called sex pilus. So through sex pilus or through a bridge, A resistant bacteria transfer its resistant DNA material to a susceptible bacteria, and thereafter acquiring this resistant DNA by susceptible bacteria, sensitive bacteria get converted into the resistant one, and therefore the bacteria become resistant. So these transferring of genetic material which is a resistant one to a susceptible bacteria through these three process and get conversion into the susceptible to the resistant one. Now we have to go through the another property that we already discussed, that is through the mutation. Another is changes in the protein of the resistant bacteria, protein in the protein of the resistant bacteria. So that is why we are calling it the altered expression of the protein of the resistant bacteria, that we are going to discuss in detail here. So what the changes or alteration occur in the proteins of the resistant bacteria, that we are going to discuss. So what will happen? These changes may be, the first one is the modification of the target site. So what is this target modification in the target site? The modification in the target site, it may be target deficient, means the number of target get reduced, that is the lack of the target, or the modification or the alteration in the target for whom? Target of the antibiotic, in where? In the bacteria. So if the target of the antimicrobial agent in the bacteria either deficient, or either its structural changes, structural modification occur, then the bacteria become resistant, are not going to be killed by the that particular antimicrobial agent, that we are going to discuss one by one. So alter target means every antimicrobial groups are possessing its own targets, that we already discussed. Take the example of beta lactam antibiotics. So beta lactam antibiotics target is enzyme. Main enzyme is transpeptidase, or other enzymes are also there. And this is called PBPS, penicillin binding protein. So when this modification occur in the penicillin binding protein or the transpeptidase, then the target of that susceptible bacteria get changed. So when the target get changed, how the antimicrobial agent can identify the target? So if the antimicrobial agent are unable to identify its target, the antimicrobial agent is not going to kill the or halt the growth of the bacteria. So that, take the example of the altered targets. Take the example of fluoroquinolones. The fluoroquinolones target is DNA gyrase. So what is this DNA gyrase? DNA gyrase is the enzyme participating in the duplication of number of DNA, means ki replication of the DNA, or if the number of DNA get duplicated or get converted into two parts, at that time this DNA gyrase is participating to inhibit the supercoiling in that DNA. So what this fluoroquinolones is going to do? This fluoroquinolones is going to inhibit the activity of enzyme DNA gyrase of the susceptible bacteria, and therefore bacteria is unable to replicate and not multiply. So bacteria will die. So what the resistant bacteria will do? Resistant bacteria modify the target DNA gyrase, and therefore fluoroquinolones are not going to identify the target, and not going to inhibit the activity of DNA garage, and therefore resistant bacteria should not be killed or its growth should be inhibited. Other example are like aminoglycoside, chloramphenicol, clindamycin, beta-lactams, macrolide, rifampicin, sulfonamide. So in these all the resistant bacteria change its target. So in case of beta lactam antibiotics, the change in the target like penicillin binding protein, that is the PBPS, penicillin binding protein. So staph pneumoniae become susceptible to resistant because this beta lactam structural changes occur in the beta lactam targets, that is the penicillin binding protein, or you can say yeah, transpeptidase changes occur. Therefore the beta lactam antibiotic agent are not going to identify the target like penicillin binding proteins or the enzymes like transpeptidase, and therefore the resistant bacteria should not be killed by the beta lactam antibiotics. Now we have to go through the other way of the altered expression of this protein. So the three ways: modification in the target, then the decreased accumulation of antimicrobial, and then the enzymatic inactivation of the drug. Enzymatic inactivation of the antibiotics. So now we have to discuss one by one. So second one we already discussed the first one. Now we have to go through the decrease accumulation. So if the target of the antimicrobial is lies or is present in the cytoplasm, drug has to penetrate or reach in the cytoplasm through the membrane, through the cell membrane. So it should be passed out through the cell membrane. So if a resistant bacteria by any means changing its accumulation in the cytoplasm, the bacteria become resistant because the sufficient concentration of the antimicrobial agent will not reach in the cytoplasm to act on the target, that is on the protein synthesis, that is on the ribosome, that is on the 50s 30 or 30s ribosome, that is on the folic acid synthesis, that is on the DNA replication, DNA synthesis. So therefore that is going to be inhibited by the that is going to be the sufficient concentration should not be there to inhibit the synthesis of protein, folic acid and DNA replication. That we have to go through in broad way. Take the example, antibiotic has to reach in the cytoplasm, should pass through the porine, and this porine is a protein present in the cell membrane of the bacteria. So this membrane, cell cytoplasmic membrane, should be penetrable by the drug. So by any means, if the structural modification in the porine occur or the structure of the porine get changed, then the drug should not be penetrate and come in the cytoplasm. So if the drug will not reach in the target site in the cytoplasm in sufficient concentration, then how it will kill the bacteria? So bacteria is either going to inhibit the accumulation of antibiotic by inhibiting the entry of the antibiotic, or by effluxing it out. by throwing it out from the cytoplasm, by evacuating it out from the cytoplasm. So by checking the entry, by inhibiting the entry of antimicrobial agent through the cell membrane, through the cytoplasmic membrane within the cytoplasm, or if the drug even though reach in the cytoplasm, the drug should be evacuated, should be ejected by the through the membrane, and therefore the sufficient quantity of antimicrobial agents should not be in the cytoplasm to check the protein synthesis, folic acid synthesis, or DNA synthesis, depending on different type of antimicrobial agent. Now we have to discuss this. So this porine, as we know, porine is the structural protein present in the cytoplasmic membrane or cell wall of the bacteria. So when the structural changes occur in the porine, antimicrobial agents should not be penetrate, should not reach in the cytoplasm, and therefore it will not act on the target present in the cytoplasm. And even though if it will reach there, the bacteria, resistant bacteria has developed some property to efflux it out, then the drug present in the cytoplasm has to be efflux out, and therefore the sufficient concentration of the antimicrobial agent is not there to kill the bacteria. That is usually with the resistant bacteria in both the cases. Take the example of both of this one. So gram-negative organism is inherent type of resistant produces, because that is going to check the penetration of the beta lactams through the porine protein, also tetracycline and chloramphenicol, but the less, because these two are also acting against the gram negative, and they are going to change the porine or structural protein. And even though it will reach there in the resistant bacteria, the resistant bacteria has developed efflux out property to efflux it out. The example is Enterobacter. Enterobacter is developed the property to efflux it out. The example is cephalosporin. So this enterobacter is going to either altered porine channel, or either this is going to this is the enterobacter is mainly changing the structure of the porine, and therefore the cephalosporin or beta lactams are not unable to enter in the cytoplasm. In case of efflux, increase efflux, this is seen with the fluoroquinolones, with the macrolide, with the tetracycline. And the last one, under the altered expression of protein of the resistant bacteria, is the resistant bacteria that we already discussed have going to synthesize some type of enzyme, and that enzyme is going to destroy the antimicrobial agent, and therefore the antimicrobial agent lost its structural integrity, and therefore the drug is not able to kill the bacteria. Now we have to go through this one example. So take the example is beta lactamase enzyme or penicillinase enzyme or cephalosporinase enzyme. These enzymes are synthesizing by the resistant bacteria, and these beta lactamase enzyme, the name is clearly telling you these enzyme releases by resistant bacteria, and these enzymes are going to destroy the beta lactam rings of the antibiotic beta lactams. The examples are penicillins, cephalosporins, and therefore beta lactamase enzyme destroy the beta lactam ring, and the drug lost its integrity to act against the bacteria to kill it, and therefore this is the property of resistant bacteria. Other enzyme that is released by the resistant bacteria are acetyltransferase, esterases. This acetyltransferase, the name is also clear cut telling the transfer of acetyl group. These enzymes synthesized by resistant bacteria, and these enzymes are participating to transfer the acetyl group to the antibiotics. So when the acetyl group is going to transfer to antibiotic, the antibiotic lost its structural or chemical integrity and lost its antibiotic property against that resistant bacteria, and the example is chloramphenicol or aminoglycoside. So acetyl group transfer by the resistant bacteria by using acetyl transferase to the chloramphenicol and aminoglycoside, and therefore these two group lost its chemical integrity, and therefore it is not going to kill or halt the growth of the resistant bacteria. Another enzyme is the esterases. Esterases enzyme are going to hydrolyze the lactone ring, and that lactone ring is the structural integrity of the macrolide. So macro means ढेर सारा बहुत सारा, macro means many lactone ring is present in the macrolides antibiotics like erythromycin, and therefore the resistant bacteria, when it releases the esterases enzyme, this enzyme is going to hydrolyze the lactone ring of the macrolide. So therefore macrolide lost its integrity to act against the resistant bacteria. So this is all about the resistance developed by the bacteria.